Hello student, in this video we discuss the unit 2 of the Mendelian genetics that contain the topic gene therapy, gene mutation and Mendelian genetics. So first we covered the gene mutation. First of all, what is mutation? What you know about the mutation? Mutation is the alteration or changes. What? Alteration and changes in gene. Any changes occur, any change in gene, that is called a mutation. Mutation may be occur by spontaneously, means instantly or naturally, maybe, or it's maybe induced type, or maybe it's duplication, deletion. There are several types of gene mutation. Here we discuss about gene mutation. Means what? Means mutation that occur in the gene. Mutation that occur in the gene, that mutation is known as gene mutation. Okay. So here we study about the mutation in the gene. So first of all, introduction. What is the gene mutation? Already I told that is mutation means a change or alteration. So here a mutation is a change or alteration okay. Mutation is a change or alteration happen in a DNA. What? Happen in a DNA. DNA means gene, right? Where DNA is located? You know that it is located in the nucleus in the gene. That DNA is our genetic material. Okay. So mutation is a change or alteration happen in a DNA, gene or chromosome due to intrinsic or extrinsic factor such as an error in replication or exposure to ultraviolet or like other factors. There are several factors that change the gene. Change in gene means here this is a DNA or this is a chromosome. Chromosome contains the DNA, right? This is our chromosome. In chromosome, this is a DNA, right? So here, if there mutation occur, if there any mutation occur in this chromosome, means mutation occur in DNA. So if there mutation occur in DNA, mutation in DNA means either deletion of the nucleotide, or addition of the nucleotide, or inversion, means there are changes the places of the nucleotide. If there any change occur in the nucleotide of the DNA, this is nucleotide, this is the DNA and this is the nucleotide, okay, that DNA content. If there any changes occur in this single nucleotide, remember, if any mutation can occur in this single nucleotide, means if T is changes to T is changes to G, C or any other nucleotide, then the gene will be changed. What the purpose of this DNA is change? Means from this DNA, RNA is formed. This is mRNA, right? mRNA is formed after transcription. After transcription, translation will be takes place, right? After translation, protein will be formed. And how this protein will be formed? By this codon, means 3 nucleotide code for 3 nucleotide code for 1 codon, right? 3 nucleotide work as 1 codon of amino acid, and this amino acid is known as chain of amino acid is known as protein, and this protein function as a enzyme or other factor. If this polypeptide or this DNA code for the protein or enzyme like insulin, and if there any mutation occur and this changes to trypsin or any other digestive system enzyme, then what happen? There is totally changes of the function. So if change occur in one nucleotide, then whole properties of that DNA or whole property of that protein will be changed. So mutation is very helpful or harmful, both can be happen through this mutation. So come here, change in the nucleotide sequence of the DNA is a mutation, right? Change in the nucleotide sequence of the DNA is mutation. The mutation is an important biological process in nature. Really, it is very important in nature, because without mutation there is no evolution of the gene, or evolution will be takes place in this nature. Only by this mutation process there is changes in the evolution. You know that there is a phylogenetic tree, right? In that phylogenetic tree we study about from single cell organisms to multicellular. How this can be possible? This can be possible only through the process of mutation. So this mutation is useful for us. What? This mutation is useful for us, not harmful. So there are harmful and useful, both property are there through mutation. Okay? The mutation is an important biological process in nature. It can be helpful or harmful. Helpful, you know. How it acts as harmful? If there is mutation, suppose this is a gene, right? And this is a gene, and their mutation occur in single nucleotide A, T, C, G, A, T, C, G. This is a nucleotide. If their mutation occur in this T, right? T nucleotide, and after mutation, the protein that synthesized that code for a cell cycling enzyme, right? That code for cell cycling enzyme. And this the property of this enzyme is what? They do not stop the process of replication. If the process of replication will not stop, means cell division continuously takes place, and there is no termination of the cell division, then what happens? Then cancerous cell will be produced. What? Cancerous cell will be produced. Many cancers are caused due to process of the mutation. Either it is by the ultraviolet radiation, or it is natural spontaneously. Maximum cancer will be caused by with the process of mutation. So it is very harmful. The mutation create variation in nature by providing new allele in nature, and hence help in evolution. I already told that in phylogenetic tree, already study from single cell organisms to multicellular, how this will be happen. This process will happen only with the help of the process of the mutation. The word mutation was similar to the French word muter, coin, which literally means process of changing. Although the mutation was originally derived from the Latin word that is mutere. The meaning is to change. Overall, the meaning of the mutation is the change, right? So here is different types of the mutation. What are the different types of the mutation? Mutation is of several types, like deletion. Deletion means there are some parts of the DNA or gene, like suppose is code for GTC, and this GTC will be deleted. Then this is known as the deletion. This is known as deletion. Duplication means here this is the gene, right? Here this is a gene, and this after replication this gene will be duplicate. How this duplication will take place? With the process of the crossing over. when cell division will takes place, when cell division will be takes place, there are the two types of cell division, meiosis and mitosis. In meiosis, crossing over will be takes place in prophase first, right? In that crossing over, crossing over, there is a sometime recombination will be takes place, or non disjunction process will be takes place. By this process, there will be happen that gene duplication will be happen, right? So here there is a duplication of the gene due to process of non disjunction. Mainly this process will be happen after the old age. In old age people, this process will be happen, duplication. Inversion means there is, if here A, T, C, G, then there will be inversion, means this G will go up and this A will become down, right? So this process is called inversion. Here there is insertion. Insertion means there are some gene will we go and insert in this. Suppose this is the chromosome 20, this chromosome 20 go and insert in chromosome 4. So this process is known as insertion. Some part of this chromosome 4 will go and insert into chromosome 20. Another is translocation. Translocation means there are two type of chromosome 4 and chromosome 20. Some part of chromosome 20 go in chromosome 4 and some part of chromosome 4 go into the chromosome 20. This process is known as translocation. Clear? So first mutation is point mutation. What is point mutation? Point mutation means there is a change in a single base of the DNA. So this is the sequence of the DNA, TA, CA, CG, TA. This is the sequence of the DNA. If there is a change in single base, means there is a change in this A, right? If there is change in this single base A, then what happened? See, this is a normal gene, TACG, and here what happened? Here from G there is a point mutation will occur. There is change in this G. Changes will occur and this guanine is changed to adenine. What? Guanine is changes into adenine. Means there is an insertion. Insertion will take place. Sorry. Here not guanine will be changed into adenine. Here one adenine will inserted before the guanine. What? Insertion means what? Addition. There is one addition of the nucleotide. So here adenine is added before the guanine. So what happened? Whole frame shift will be changed. Frame shift of the protein will be changed. And here what? Here methionine, arginine, threonine, phenylalanine. This gene code for methionine, arginine, threonine, phenylalanine, and if insertion will take place, then this gene code for methionine, serine, tyrosine, leucine. Whole protein will be changed. Then the function of the protein will be also changed, right? In deletion process, what happened? Here there is a deletion of the one nucleotide. What? Deletion of the one nucleotide. Means this guanine will be deleted. See, here this guanine will go away, means guanine will be deleted. After deletion of this guanine, frame shift of the nucleotide will be changed. And this after changing, this code for methionine, valine, proline, and here some other codon. So what happened? Here also the nature of the protein will be changed. So after adding, insertion or after deletion, after insertion or deletion by both process, there is a change in the property of the protein. So this is a very harmful process. When a single nucleotide deletion means when a single nucleotide will be removed. Insertion means when a single extra base is added into the codon. This is known as insertion. Next mutation is silent mutation. Silent mutation means in codon there are 22 amino acids, right? You already know there are 22 amino acids. 22 amino acid coded by 61 codon, right? Means there are several codon which like two codon code for 1 amino acids. Some are there are 3 codon that code for 1 amino acids, right? So in silent mutation what happen? If a mutation will takes place, means their insertion or deletion of codon will be takes place, right? After inserting or deleting the nucleotide, after that codon that change a point mutation will occur. That codon code for same amino acids. Means there is no changes in the wild type 1. Then that mutation will known as a point silent mutation. Means mutation will take place but there is no change in the enzyme or protein in polypeptide. There is no change in the property of the polypeptide. Then that type of mutation is known as silent mutation. See here, this is a DNA, right? This is the DNA sequence. Here what happened? There is a change in the T amino acids. What happened? There is change in the T amino acids. This is the TTC. What? This is a TTC. This TTC code for after transcription, this code for AAG, and this AAG code for the lysine, code for the amino acid lysine, right? After mutation, point mutation, this C will change to T. Mutation will take place at the place of C and that converted into T, thymine, right? And this thymine after transcription code for triple AA, right? And this triple AA code for the amino acid lysine. Means there is no changes in the amino acids. If there is not change in amino acids, means the nature of the polypeptide will be same as the wild type 1. So this type of mutation is known as silent mutation. This type of mutation is known as silent mutation. Okay, next is our nonsense mutation. Nonsense mutation means when we add or alter the nucleotide, that nucleotide code for a stop codon, right? If that nucleotide code for a stop codon, then that is known as nonsense mutation. A stop codon is added. A stop codon is added to the premature protein. It is stop protein synthesis because a stop codon ends synthesis of protein and result in a premature protein or truncated protein. When a stop codon is added, means there is a premature formation of the protein. So that type of mutation is known as nonsense mutation. Okay, means termination will be takes place after the addition or after the deletion or there is a mutation in a gene that code for the stop codon. there are 64 codon. In that 61 code for amino acids and rest 3 code for stop codon. Okay. So nonsense mutation is a addition of stop codon that terminate the translation of the protein, right? Next is missense mutation. Missense mutation means if there is changes will occur, mutation will occur in a nucleotide, and that nucleotide code for other type of amino acids. Means there is mutation will occur in middle position, right? Mutation will occur at middle position, and this mutation will change the mRNA, and then this mRNA codon code for arginine. What? Arginine means other type of amino acids. Means this amino acids will be changed, and this after changing the amino acids, whole the nature of the polypeptide will be changed. So in missense mutation, a codon originated from a nucleotide change that will code for different amino acids. It can lead to alteration or loss of a function of a protein, right? If there is a change of the function of protein will be occur, or there are a loss of the function of protein will be occur. Okay, right? This is clear. Next is induced mutation and spontaneous mutation. What is an induced mutation and what is a spontaneous mutation? Induced mutation means if there is alteration of the gene occurred by the process of the radiation or by the process of the chemical agent or physical agent, then that affect the dideoxynucleic acids molecule of a living organism. This type of mutation is known as induced mutation. And spontaneous mutation means if there is mutation arises naturally. What? Naturally. That type of mutation is known as spontaneous mutation, right? So what is induced mutation? If mutation is occur by a physical or chemical agent, that type of mutation is known as induced mutation. And a mutation that occur by a natural agent, not by a exposure to a mutagen, that is known as spontaneous mutation. Induced mutation occur due to mutagen present in the environment. Mutagen like ethidium bromide, ultraviolet radiation. This type of mutagen is known as this type of mutagen are present in our environment. Occur due to the incorporation of base analog, base mispairing and base damage. This mutation, induced mutation, occur by when base analog are substitute at the place of the nucleotide, or there are a base mispairing will be happen. This type of mutation is known as induced mutation. This mutation are most important in where? In reverse genetics. What? In reverse genetics. And spontaneous mutation are important in a forward genetics. Induced mutation occur due to the natural causes, not by the mutagen. Occur due to errors in DNA replication, spontaneously lesions like depurination, deamination, transposable element like this. This type of mutation is a spontaneous mutation, right? Next is what is gene therapy. Gene therapy is a process. What? It is a process or we tell as techniques by which we cure or improve the mutated gene or a gene that cause a disease, we treat that gene is known as gene therapy, is known as gene therapy. This is a therapeutic transgene that grow into the nucleus. This is the target cell, go inside the nucleus. Here this is a therapeutic protein will be formed, and this therapeutic protein will cure that gene. So this technique is known as gene therapy. Gene therapy introduction. Gene therapy is a novel treatment method. What? This is a new technique. What? This is a new technique which utilizes gene or short oligonucleotide sequences as therapeutic molecules instead of conventional drug compound. This technique is widely used to treat those defective genes which contribute to disease development. Mainly this gene therapy is used to disease developing or defective gene. This is our gene. This package the healthy gene RNA interference or gene editing tools into the AAV. Right? We package this gene into a AAV. Inject the package AAV into the tissue of interest. We inject this AAV into the tissue of the interest. After going inside the tissue of the interest, this AAV will enter the cell. This AAV enter into the cell and release the genetic material. The cell will become healthy by either expressing the normal gene. This actually, what is this? This is a healthy gene, right? This healthy gene, we convert this healthy gene into what? AAV. After conserving, we inject this gene, right? We inject this gene into a target tissue. Our target tissue is our brain. Then we inject into the brain. This gene go into our brain. What happened there? There are three possibilities. Either this gene translate there and function as normal gene, or this gene replace the mutant RNA, right? This gene replace the mutant RNA, or this gene correcting the mutant gene. This correcting the mutant gene. So in all this process, in all these three possibilities, the defective gene will be correct. The defective gene will be correct, right? So gene therapy is the method by which we correct the defective gene which contribute to the disease development. Next is type of gene therapy. There are two types of gene therapy: somatic cell gene therapy and germline gene therapy. In somatic cell gene therapy, this type of gene therapy will take place in the somatic gene. Therapeutic genes transfer into the somatic cells. That is known as somatic cell gene therapy. And if therapeutic gene transfer into the germ cells like egg or a sperm, if that gene transfer into the egg or a sperm, then this is known as germline gene therapy. Example: introduction of gene into a bone marrow cells, blood cells, skin cells. This is known as somatic cell gene therapy. Will not be inherited later in generation. At present, all research is directed to correct genetic defect in somatic cells. Means somatic cell gene therapy will not inherited or not transferred to generation to generation. Why? Because during gamete formation or during zygote formation of the zygote, only germ cell will act as carrier of the information, not somatic cells, right? So if there is any alteration or any changes will take place in the somatic cells, then that information will not transfer or not inherited in later generation. Whereas in germline gene therapy, it is heritable and passed on to later generation. Why? Because this zygote will be formed by this sperm and egg, and this is zygote contain that therapeutic gene therapy information. So that information will be passed generation to generation in later generation, right? So germline gene therapy is a heritable, okay, whereas somatic gene therapy is not to be inherited. There are two types of gene therapy approach. First is classical approach and second is non-classical approach. In classical approach, therapeutic gene delivery and their optimum expression occur once inside the target cells. The foreign gene carry out of the following function. What type of function will be carried out by the foreign gene? Foreign gene will produce a product or protein of that type of protein which patient lack. This is a patient. Suppose this A is a patient. This A patient lacks some protein, like they lack insulin protein, right? Then this foreign genes go or carry that protein, means insulin protein, and they transfer into that patient A, right? Second, produces toxin so that disease cell will be killed. Third, activates cell of the immune system so as it help in killing the disease cell. Overall, our target is what? Overall, our target is to stop the or cure the defective gene which cause the disease, right? So in classical gene therapy, the foreign gene will cure the patient by killing the disease cell, or by activate the immune system, or produce a product that they lack. Okay. Another is non-classical gene therapy. In non-classical gene therapy, there is an inhibition or expression of the gene associated with the pathogenesis or to correct a genetic defect and restore the normal gene by the process of there are two type of process. First is ex vivo gene therapy and another is in vivo gene therapy. In ex vivo gene therapy, there is transfer of gene into a patient through outside the body, and in vivo there is transfer of gene directly to cell inside the body. Okay, inside the body there are two type of gene therapy, right? So this is the diagram of the in vivo and ex vivo. In vivo, this is a patient, right? This is a patient. This is a therapeutic gene. We isolate that therapeutic gene and package into the virus, that is act as vector. Virus act as a vector. This after transferring into packaging by the virus, then inject into the patient. After injecting into the patient, that patient cured by that defective gene, right? In ex vivo, we isolate the stem cells from the patient, and there is a therapeutic gene, transfer that therapeutic gene into the virus, and we transfer both these stem cells and this virus containing therapeutic gene into a virus transduction in the lab. And after this treatment of the cells, we again inject into the patient. Okay, these are the two type of non-classical gene therapy. Next is Mendelian postulates. Mendel is the father of the genetics. First, the genetics term are come from the Mendel. Actually, he study the trait in the field. Actually, he is a farmer, and during their work, he studied the gene. That time he called that gene as a factor. Study trait that occur in a distinct form, develop true breeding varieties. When breed amongst themselves, this plant produces offspring identical to the parent or that trait. They use mathematical analysis in the study. They study in the pea plant. What? Pea plant, Pisum sativum. And they select 7 characteristics. 1, 2, 3, 4, 5, 6, 7. This is the characteristics of the pea plant that Mendel take. He take pea plant because they easily grow and there is less time duration of their growth period. So he select the pea plant. Okay. Second, modify to incorporate today's vocabulary like genes, alleles, because Mendel called that gene as a factor that time, right? First, that factor he called there is a substance, there is a factor that take all the information or that transfer the information from generation to generation, and that is known as a factor. Nowadays, that factor is called as a gene, right? The hereditary information that determine a single trait. Allele is alternate form of a gene. Every gene have a 2 form. That alternate form of gene is known as the allele. When an organism inherits 2 identical alleles for a trait, organism is said to be homozygous, or they are inherit different type of allele, then that is known as a heterozygous. See, this is a homozygous, means they are transferring same form of allele, and this is a heterozygous, means they are forming a different type of allele, right? So this is a homozygous versus heterozygous. Next, test cross. Actually, what happened in test cross? Test cross are used. Test cross are used to identify or determine the genotype of an individual. If you want to know the genotype of the individual, then you use the process of the test cross. Test cross is a type of back cross. Means if there is offspring, if you want to identify the genotype of that individual, we cross that individual to their parents. What? Cross that individual to their parents, then we determine the genotype of that individual. This is known as test cross. This is a homozygous tall, this is a homozygous tall, this is homozygous dwarf. When we cross this both, then the heterozygous tall will be formed. You want to determine the genotype of this all tall. What happened? They cross with any type of the any parents of any parents. Cross this parents, this tall, heterozygous tall, cross with homozygous dwarf. Then what happened? Tall and dwarf both will be there. Means what you think? Means this offspring are heterozygous in nature. Remember one thing, in test cross, we cross with only recessive parents, right? Not with dominant parents. Whereas in back cross, we cross with both in recessive and homozygous dominant both. Next is chromosome mapping. What is a chromosome mapping? Chromosome mapping is a genetic mapping. we call as also called as genetic mapping or genome mapping, because chromosome is our genome that contain our genome and we map the genome. This is process known as genome mapping. Genetic mapping is based on the use of genetic techniques to construct a map showing the position of the gene and other sequence feature on a genome. Right? Actually, chromosome mapping is done to identify the location or position of the gene. Suppose we want to identify the Gaucher disease or glaucoma gene. Okay? What happened? How can you determine this? This will be determined with the help of chromosome mapping. We map the chromosome. Right? Genetic techniques include cross-breeding experiment or case of human examination of family histories. Right? Next is physical mapping. Physical mapping uses molecular biology technique to examine DNA molecule directly in order to construct map showing the position of sequence feature including the gene. Linkage is a method by which actually it is a linkage a recombination method by which we determine the frequency of the recombination in the gene, or they are linked gene or they are unlinked gene. There are two type of gene. Linked, if linkage will takes place means they are linked gene. If linkage is not takes place means they are unlinked gene. There are two ways in which gene may be unlinked. They may be on the separate chromosome. They may be far apart on the same chromosome. If two genes are far apart from each other, means linkage will not take place. If they are closely associated with each other, then linkage will take place. Right? For linkage, the distance between two genes will be less. Only after that linkage will be takes place or crossing over will be takes place. Okay? Linkage will takes place with the help of crossing over. And linkage will be takes place always less than or equal to 50%. Okay? Less than or equal to 50%, not more than 50%. Remember this. The recombination frequency is always less than equal to 50%. Right? So here this video is about the gene therapy, gene mapping and mutation. So here we stop here. So thank you.